Hello everyone. Today I brought with me an air balloon. The most ordinary air balloon you ever seen. And I would like to use this air balloon to enhance your vision. Actually to enhance everyone's vision. And I promise it's not going to hurt. Are you ready? Yeah. Can you see what's written here? Probably not. Very well. Let's blow up this balloon and see what's gonna happen. So balloon is expanding. The letters getting bigger and bigger and bigger, and now you can clearly see and read this word. It's written synapse. It's one of my favorite words, and it's alright if you don't know the meaning of this word. We're gonna talk about it a bit later. So making small objects bigger is an interesting concept, but is it a practical one? As you can probably guess, unlike air balloon, it's hard to blow up solid things, such as, for example, my hand. We're not getting bigger, right? So how can we get small things bigger without violating any basic laws of physics? Unless we're in a sci-fi movie, it's really hard to break laws of physics. But I promise we're gonna break one later today. Let's carry out another experiment. For this, I got a baby diaper with me. So in its initial state, the baby diaper is thin. And now let's pour some water on it. Here I got the water with me. I always label my chemicals. Many babies around the world do this experiment every day. And maybe you did this experiment many times too when you were a baby. And also if you decide to repeat this experiment at home, I recommend you to use a fresh diaper. So it takes a little bit of a time to absorb this water. Usually babies don't pee half a liter in one go. A few more seconds. And as I add water, you can see this baby diaper is expanding. Do you see it? And here is the negative control. The baby diaper is expanding because it contains a super absorbing polymer. Same polymer that you can find. The baby diapers contain same polymer as you can find, for example, in water expandable toys. So polymer consists of a long chains of atoms that are collapsed into each other, forming dams. And if we can zoom in and see the molecular structure, we will see the long chains of atoms that are collapsed to each other, forming dense networks. And the distance between molecular chains are similar to the size of the average biomolecule in our body. And when we add water, water molecules get in between these chains through a process called osmosis and push them away. So the polymer expanding up to 1,000 times in volume. Since the polymer chains and distance between them are comparable to the size of the biomolecules in our body, can we use this process to blow up biological samples on a molecular level so you can see the cells and molecules they consist of better? Surprisingly, but it can be done in 4 simple steps. So step number 1, we take the sample, for example, a brain tissue or a neuron, and we infuse the building blocks of a polymer, monomers as we called, inside. Then we initiate a chemical reaction, polymerization. During the polymerization, biomolecules get incorporated into the polymer chains. And these polymer chains find their way around, between, and within the biomolecules. So this eventually will help to pull them apart. Then we need to do one more step. We treat the sample with a special chemical that will destroy the interactions between the biomolecules so they don't resist to expansion. Then we add water, polymer will start to swell, the biomolecules will start to go apart together with the polymer chains. And in my lab at Westlake University, we use this technology, which we call expansion microscopy, to make things bigger, such as brain. But what's going to be happening when we embed the brain cells into the gel and we start to expanding? They're going to be bigger, up to a 6-fold, to a point where you actually can see it with the naked eyes. It's amazing. You can see cells with the naked eyes after expansion. However, at the same time, you can ask, why going through the troubles of making things bigger if you can simply take a light microscope? And this is how the neurons were discovered in the first place anyway. However, all conventional light microscopes have a fundamental limitation. Light propagates as a wave, similarly to the wave on the surface of the water. As a result, light does not always travel as a straight line. It can bend around the corners of an object, the phenomenon known as diffraction. This is one of the reasons why, for example, the shadow casted by an object does not have a crisp edges. Similar thing happening in the microscope when the light passes through an objective lens. It's getting smeared out and diffracted in the focal region, and the spot of light that can be formed will be at least 200 nanometers in size. In other words, if there are two objects with a position closer than 200 nanometers to each other, will not be visible in a microscope. We won't be able to tell them apart because they're gonna appear as a single blur under the microscope. It means that the microscopes cannot be used to study biological structures which are smaller than 200 nanometers in size. And this is synapse is one of the structures which is smaller than 200 nanometers in size and thus is not visible under optical microscope. But what is the synapse and why do we want to see it? Synapse is a bridge between two neurons in our brain that transmit information from one neuron to another. By passing information through the synapses, our brain generates everything we think, feel, sense, and do. And if we ever want to understand how our brain works, we need to see and map every single synapse in our brain. So expansion microscopy is one of the technologies that help us to physically magnify the brain and let us see single synapses under conventional microscope. This makes expansion microscopy a technology that opens a window into a mysterious world of our feelings and thoughts and memories and creativity. At the same time, it let us appreciate the brain complex structure. In a cubic millimeter of our brain, there are 100,000 of neurons connected by a billion of synapses. And I believe one day an army of microscopes will image and capture every single synapse in our brain. And then AI-controlled supercomputers will go and analyze these images and make a comprehensive wiring diagram of our brain. Seeing the wiring diagram of our brain will not only help us to answer the long-standing philosophical question of who we are, but we also will have tremendous practical utility. It will help us to cure diseases. It will create new brain-machine interfaces. It will also inspire new architecture of artificial neuronal networks toward general artificial intelligence. Until then, imaging and mapping every single synapse in our brain remains the most technologically challenging task of all times. It will take a work of generation to succeed. And still, I believe, I will live to see that day then a substance from a baby diaper will help us to solve a brain. Thank you.